Mast cells are most well known for their inflammatory response in allergic reactions. There is also evidence that suggests that their inflammatory response is responsible for the flushing out of parasites such as nematodes and hookworms. What is less known, though, is what role mast cells play in the brain. Mast cells are one of the only residential cells of the brain. However, systemic mast cells are also able to travel in and out of the brain. Some researchers have considered that behaviour may be linked to mast cells in humans, with one recent study linking a deficiency of mast cells to anxiety-like symptoms in mice. They tested mast cell deficient mice against wild-type mice in a number of anxiety tests, with the mast cell deficiency mice scoring higher in each of these. I would like to look further into this, adding mast cells to mast cell deficient mice to see if this reduces their anxiety-like symptoms, as well as testing if there is a difference between mice with mast cells only in their brain, in their body, or in both. Let's have a look at the experimental design.
For this experiment, we will have 5 groups of mice. Group 1, which is being used for cell isolation and culture, will have 10 adult mice and 10 baby mice. Groups 2 to 5 will each have 10 adult mice. In Group 1, which is being used for cell isolation and culture, mice will first have their serum extracted from their body. They will then be decapitated, their brain collected and blended so that we can collect a sample from the brain. The cells will then be centrifuged, pelleted, and washed before being incubated in rabbit anti-Fc epsilon R1 gamma antibodies that will attach only to mast cells. They will then be incubated in an anti-rabbit antibody that will attach to this first antibody and is also attached to a magnetic bead. The samples will then be sent through a magnetic chromatography tube. This is a very specific type of chromatography because only cells with the magnetic bead attached, in our case the mast cells, will attach to the side of the tube. At this point, the adult cells will be counted for future reference. The baby cells need to be cultured. This will be done in a methylcellulose medium with a mixture of IL-3 and IL-4 added. This will allow the proliferation of the baby mast cells. These mast cells will later be used for the injection into the mast cell deficient mice. In Group 2, mice will first undergo tests for indicators of anxiety. In the open field arena test, it will be measured for how long and how often mice go to the center area of the arena. Mice that tend to stay near the edges of the arena are those that have high indicators of anxiety. In the elevated plus arena, it'll be measured how often and for how long mice explore the open arms of the maze. Mice who tend to stay in the closed arms of the maze are those with high indicators of anxiety. In both these previous tests, the defecation rate will be compared to that of its home cage. Mice that showed higher defecation rates in these 2 previous tests will show higher indicators of anxiety. After the tests, the mice will have a saline injection into their brain and into their body. This will prove that our later results are not only because of the injection itself, but are because of the mast cells. After the injection, mice will undergo the anxiety tests again at 10, 15, 20, and 25 days. In Groups 3, 4, and 5, the mice will also undergo the tests for indicators of anxiety. They will then have the mast cells injected to them. In Group 3, the brain, in Group 4, the body, and in Group 5, both. After this, they will also undergo the anxiety tests again at 10, 15, 20, and 25 days. So what do we expect from these experiments? Well, if we expect that mast cells in the brain are more likely to influence behaviour, we would expect Groups 3 and 5 to show a reduction in anxiety-like indicators. However, since mast cells from the body can also travel in and out of the brain, we may also see a reduction in symptoms in Group 4.